So a very warm welcome and good afternoon to our next BIOCTANE webinar. I'm honored and pleasured to see you all on board today for this topic, as you probably know, which is biofilms on active substrata. And probably you know that the BIOCTANE process studies a pathway to produce aviation fuels from renewable sources using acetoine and 2,3-butane diol as platform molecules. And during the last webinar, we covered the thermochemical conversion routes towards those platform molecules. And today we'll be leaning further towards the biological side. And for this part, I'm extremely happy that Professor Gescha has joined us. He's a renowned expert in the field of using biological conversion pathways and the utilization of biofilms. So without further ado, Professor Gescha, the stage is yours.
Yeah, thank you so much. Let me just share my screen. Bring it to presentation mode. That should work, right?
Yes, looks great.
All right. Yeah, thank you very much for the invitation. Thanks for having me. The BioOctane project is, in my eyes, a blueprint for the majority of future projects dealing with the sustainable production of compounds, chemicals, commodities, and so on and so forth, because it nicely connects biology and chemistry. And what I will try to do is today, I will try to make you familiar with the word biofilm and with the science behind biofilms. And also with the term active substrata and what this means. And what you see here on the first slide are three examples of active substrata. And I will cover these active substrata actually also in the upcoming slides. And what you see here are biofilms that grow on surfaces. So it's bacteria that grow on surfaces. And on the left-hand side, let me switch you to laser pointer. On the left-hand side, you see a biofilm growing on a cathode. It's supplied by energy from a cathode. In the middle, you see something that we deal with in the BioOctane process, which is a biofilm growing on a membrane fiber. And then last but not least, on the right-hand side, you see a biofilm growing on an anode surface. So biofilms are usually made out of microorganisms. And because I thought that maybe some of you might actually be chemical engineers and not that familiar with microorganisms, I thought I'd just start by saying, what are microorganisms? And what you see here is actually an animation, and it shows you a nail between two fingers. And with more and more magnification, we get closer to the surface of the nail here. And what you see with 10-fold magnification, you see a hair lying on the tip of the nail. If we go to 100-fold magnification, we see actually dust mites. When we go to 1,000-fold magnification, we would see pollen, and we would see red blood cells and white blood cells. And only if we go up to 10,000-fold magnification, so we see here this scale bar is 2 micrometers, we see the organisms that are actually the unseen majority. We see bacteria, and in this case, this is supposed to be an E. coli cell, this is supposed to be a staphylococcus cell. So these organisms are micrometer in size, and they are very simple with regards to their overall structure, but they are, these microorganisms, these bacteria, are the unseen majority on our planet. And what you see here is the tree of life. This is the latest version of the tree of life. What you see here is how all known organisms on our planet are related with each other. And there are 3 kingdoms in this tree of life. This kingdom here is called bacteria, and you see that it's by far the most diverse. So the majority of organisms on our planet are actually bacteria, and these bacteria fill all the niches on our planet. And then we have the kingdom of eukaryotes, and we are actually part of this kingdom. Fungi, animals, and plants are part of these eukaryotes, and you see this is by far less diverse. And then there's the third kingdom, which is called archaea, and these organisms resemble in their shape and general structure also the bacteria. So these microorganisms that make up biofilms, they are the most diverse kingdom. They are the majority of organisms on our planet. And most bacteria, most microorganisms on Earth grow in the form of biofilms. So what is a biofilm? You all know biofilms. These biofilms is the coverage on stones that make these stones slimy and slippery when you go into a stream or into a lake. These biofilms form on the surface of your tooth. These biofilms can cause problems in terms of infections when you are in the hospital. But nevertheless, the majority of microorganisms grows in the form of biofilms. This is their natural lifestyle. But when we think of biotechnology and sustainable bioprocesses, we basically ignore this, or ignored this for a long time. And we operate all these biotechnological processes, or most of the biotechnological processes, in stirred tank reactor, and try to bring these organisms into a planktonic life form so that they grow solely in a liquid medium, and then we want them to operate. Although their natural form of growth is to grow in the form of a biofilm. And we actually would like to change this, to change the way how we think about biotechnology, and we would like to make a contribution to using the cells in their natural life form, because this natural life form comes with a number of advantages. But before I come to the advantages, let me first say a few general words more with regards to biofilms. So these biofilms are highly complex, and you get a glimpse of the complexity when you have a look here on the left-hand side. What you see is a multi-species biofilm. And in this multi-species biofilm, we have the organisms, and the organisms form something which we call EPS, extracellular polymeric matrix. So the organisms attach to a surface, and then they start to grow and they start to engulf themselves in a polymeric matrix, which consists out of sugars, DNA, cellulose, and so on and so forth. So it's not only diverse with regards to the organisms which might be here, but it's also diverse with regards of the structure of the polymers and the chemistry of the polymers in these EPS. Schematically, we have either single organisms or flocks of organisms that would attach to a surface. Then these organisms would grow, build more of this extracellular polymeric matrix, and then at some time they would start to disintegrate, to detach, either in the form of single cells or as clusters, and then they would aggregate and attach to another surface, and so on and so forth. So this is the usual life cycle of microorganisms growing in the form of biofilms. So why do we think that biofilms have an advantage with regards to biotechnology? So advantage one is, so what you see here is a surface, and we have a biofilm growing on the surface. And one clear advantage is that the biocatalyst actually sticks to the surface naturally. So it's easy to operate biofilm systems in plug flow. So we have a substrate coming from one side, and we can operate the process in a way that in the end, we have a stream that contains only the product. And we do not have to invest largely in separating the product from the biocatalyst. These cells in biofilms are usually highly resilient. They tolerate higher concentrations of substrates. They tolerate higher salt concentrations. And they tolerate substrates that are toxic for planktonic cells. Also, if we operate the process as plug flow, then there will be a stable gradient of substrate along the biofilm depth. So this typically up to 400 or 500 micrometer thick biofilm that will build on the surface, the cells in the different layers will always see one particular substrate concentration and one product concentration. And biological biocatalysts can learn, they can adapt to substrate concentrations, and they can get better over time if the process is stable. This is basically the concept of evolution. The only difference with regards to microorganisms and for instance, humans, is that we deal here with a huge number of organisms, and evolution occurs rapidly within days or at least the most weeks. So if we have a continuous bioprocess that is operated with biofilms, the organisms will adapt over time. They will accumulate mutations that are beneficial for the process if the process is designed in the right way. And the biocatalyst will get better over time. And last but not least, we have active substrata. What does active substratum means? I covered it slightly already in the beginning. So an active substratum is a surface on which the biofilm grows, which supplies the cells with a substrate. And this substrate can be, for instance, a gas like CO2, carbon monoxide, or hydrogen, or it can be an electrode that takes up electrons from the metabolism, or it can be a cathode that brings in electrons for the metabolism. And especially if we are talking about gaseous substrates like hydrogen and CO, these active substrata can be highly beneficial because these gases are not very soluble in water. CO2 might be, but hydrogen not. And it takes a lot of energy in order to bring these gaseous substrates like CO or hydrogen into solution. And if we supply these gases to the biofilm via aid of a membrane, so the gases diffuse through a membrane into the biofilm, we can have a depletion of these gases while they migrate, while they diffuse through the biofilm. And we do not have to invest in energy in order to steer the media so that we can bring as much hydrogen or CO into solution as possible. So I will talk today about 3 examples of active substrata that are used in biotechnology and wait a second. Okay. 3 active substrata that we can use in biotechnology. I will talk about the active substratum cathode, the active substratum anode, and last but not least, hollow fiber membranes as active substrata that supply biofilms with gas that diffuses through the pores of the membrane. And there are applications for all these 3 different active substrata I will talk about. You will see in this lecture a lot of optical coherence tomography data. Optical coherence tomography is our workhorse with regards to analysis of biofilm structures. And it's very important to be able to steer the architecture of the biofilms. You don't want these, or you at least would like to know what is the maximum of biofilm thickness that still allows you to have optimal process kinetics You would like to know when the biofilm gets too thick so that you have mass diffusion limitation. You would like to know what the structure of the biofilm is, whether it's porous or not, and how the porosity of the structure can actually change the process kinetics. So in a lot of projects, we start the projects with using very robust microfluidic technologies, and these are two examples of this. So this is a so-called bioelectrochemical setup. So here is a microfluidic chip. The chip has a volume of roughly 300 microliters. In the middle of the chip, this black thing that you see here is an electrode, and the biofilm will grow on the electrode. And we have a flow of medium going from one side to the other. And since this is a bioelectrochemical system, we do not only need a working electrode, but we also need a counter electrode, and this counter electrode comes then downstream of the working electrode. And when we talk about biofilms growing on membranes, then we have these single fibers of a membrane bundle that we integrate, or that my PhD student Carmen is actually able to integrate into these microfluidic structures. So you need really calm hands in order to do this. So, and if you're able to do this, if you're able to take small pieces of active substrata and integrate them into these microfluidic structures, you can then use optical coherence tomography in order to analyze biofilm growth on these structures. And we have developed two robotic systems in order to do this. So these OCT devices, they are set up on a robotic arm. We can, and this is what you see on the left-hand side, we can bring them right over the specimen and at arbitrary positions, and we can also automate this overall process when the robot is taking samples and so on and so forth. And then we get a three-dimensional impression of the biofilm structure. We see void spaces, we see how thick it is, we see what the three-dimensional architecture is, and we see this in a resolution of roughly 8 micrometers. And then we have a second robot. This is what you see on the left-hand side, and this second robot is also equipped with a sampler. So we can first make an image with optical coherence tomography, see where the biofilm builds, how thick it is, and then we can go in with a needle and take microliter samples from the biofilm and then analyze these samples, for instance, by omics technologies. We can analyze what are the genes that are active in the organisms, what are layers of organisms that are more active than others, and then we can ask why this might be the case. So the first example that I would like to discuss with you are biofilms grown on anodes. In the case of an anode, we have a biofilm that grows on a solid state anode. We feed this biofilm with an organic carbon source. And what these organisms do is they oxidize this carbon source, they produce CO2, and the electrons are then transferred onto this solid state anode. It's a special kind of respiration. So what we do as humans, we take up organic carbon, we oxidize the organic carbon to CO2, but we respire oxygen and we reduce the oxygen to water. These organisms grow under anoxic conditions in the absence of oxygen, and they use the anode as respiratory electron acceptor, and they reduce the anode with electrons. So they basically catalyze the direct conversion of chemical energy into electrical energy. How, why, and where could we use this? There are a number of rather cheap carbon sources, for instance, organic carbon in wastewater. And we can use these organisms to convert these organic carbon into CO2. We take the electrons, and if we add a little bit of extra power, then we can produce hydrogen at the counter electrode. So actually we need 50% of the power that normal water electrolysis takes in order to produce hydrogen if we use these bioelectrochemical systems here. And this enables us to use organic carbon, for instance, in wastewater that and that occurs in tons and tons and tons, and oxidize it to CO2 and produce hydrogen from this. So anode biofilms are clear examples of counter-diffusional systems. So on the one side, we have the substrate coming into the biofilm, and this substrate is depleted over the thickness of the biofilm. And then on the other side, we have the kinetics of electron transfer from the anode to the organisms. And the further away the organisms are, the least efficient is the electron transfer from the organisms to the anode. If the biofilms are rather thin, we will actually see that most of the cells can directly interact with the anode. If the organisms get rather thick, up to 400 micrometers, we see that actually only the outer parts of the biofilms are active, and the parts that are the remaining parts might only be something like a conductive polymer that the organisms here at the outside use in order to transfer electrons to the anode surface. And we have a number of projects in which we try to use waste carbon sources and would like to oxidize them to hydrogen, because we would like to use the hydrogen for further biotechnological processes, or we would like to use the hydrogen as platform chemical as it is. So I bring you here an actual experiment from the lab. This is from a lab seminar a couple of weeks ago. And this is work of my PhD student, Mashid, who is working on a project which tries to realize a wastewater treatment plant 3.0, where we actually get hydrogen from the organic carbon, which is soluble in wastewater. So what she's using is she's using these microfluidic chips. Here is the electrode. And this is the system. The organisms grow on the electrode. They take organic carbon. They produce CO2. And at the counter electrode, we produce hydrogen. And what we always would like to do is, what do we have to do in order to achieve the highest possible current density? Current density means how much electrons do the organisms transfer to the anode surface area. So, and we did a very simple experiment. We had a flow rate of 4 milliliters per minute in this small chip, 16 milliliters per minute. So with 4 milliliters per minute, we have less current production. Current develops not as fast. If we start with 16 milliliters per minute, we have more current, and current actually develops rather fast. And then we ask, well, do the organisms in the biofilm, do they actually have something like a memory? If we change the flow rate from 4 milliliters at day 4 to 16 milliliters, what will happen? And what happens is what you see here in the gray bar. As soon as we change the flow rate, the current density actually increases up to values that we saw when we also started with 16. But even more interestingly, if we start with 16 milliliters per minute and then we change the flow rate to 4, even here the organisms seem to remember the biofilm status that is most efficient compared to these 4 milliliters per minute. And if we then analyze with optical coherence tomography how this biofilm looks like, what we see is that all the biofilms that operate well in this system have one in common, and this is low porosity. And this is what you see here. All the biofilms that operate best have a low porosity compared to the biofilm that was operated with 4 milliliters per minute, over the whole time course of the experiment. So this means we can train the organisms to build a less porous biofilm, and that the less amount of pores that we have in the biofilm, the more efficient is electron transfer to the electrode surface. And if we build larger scale reactors and we try to follow this rule of having this kind of shear stress on the organisms, on the biofilm, we can actually get very efficient processes. Another way to increase process efficiency is not to use natural biofilms, but synthetic biofilms. And what we do in terms of synthetic biofilms is that we spray biofilms with a very simple spray gun on the surface of, for instance, an anode. And if we do this in a hydrogel, we can actually also functionalize these hydrogels so that it contains, for instance, carbon nanofibers, which are functionalized with riboflavin, an organic redox shuttling molecule. And if we do this, if we use these functionalized hydrogels as basis for a spray coating of surfaces with biofilms, we see that compared to the natural system, we get an increase of roughly tenfold in current density. So there are two ways in order to try to optimize these systems. Either we just go with process conditions, or we actually try to build synthetic biofilms from scratch and build these processes. In terms of upscaling, if we want to upscale these systems, especially if we want to use them in wastewater treatment, we can learn from conventional wastewater treatment plants. And we did this in the past. So what you see here is an example of a so-called biological contactor, which is already in use in wastewater treatment plants around the world. And what these contactors do is they turn very slowly in wastewater, and they bring this water up and then down. And we have a biofilm of organisms growing onto these discs here. And by rotating these discs out of the water and bringing them back into the water, we supply the organisms with sufficient amounts of oxygen that cannot diffuse well into the wastewater. And we thought that we can actually do the same thing also with bioelectrochemical systems. And we set up this reactor setup here where the rotating discs are actually the anodes of our system, and we have the counter electrodes, the cathode, just a centimeter away from these systems. The discs turn around with a motor, and we can increase motor speed, thereby increase shear force and compress the biofilm. And we also have like a mixing vessel in order to adjust pH. And we did experiments with this in 10-liter scale, and we also built these reactor systems in collaboration with colleagues from the KIT to 100-liter scale. And these are experiments in 10-liter scale where you actually see that we can monitor current density here in black over time. The systems give us maximum current densities with waste substrates of up to 1.5 amps per square meter. And we can actually also see what the biofilms like the most, what is depleted the most, which is in this case acetate, a very simple carbon source. And another thing we can do is we can actually steer the biofilms. We can, by adjusting the potential of the electrode, we can also adjust the current density. So we can tell the organisms how fast they should respire under certain process conditions. And we can use this in order to increase or decrease the overall voltage of the bioelectrochemical system. We did an upscaling experiment, a real upscaling experiment at the last flower show in Germany, Bundesgartenschau, which was held in the middle of Germany in 2023. And here with Harald Horn, who you see on the picture, we built a toilet for the visitors of this Bundesgartenschau. And here you see what is actually in the center of this little building. Here is a 100-liter bioelectrochemical system. We have all these rotating discs in the middle. We measure hydrogen. We have a potentiostat in order to adjust the potential. And these three guys here are very important in this time period of 9 months in which the system operated. Next example of active substrata membranes. So in these membrane biofilm reactors, we have these membrane fibers, and on the surface of the membrane fibers, we have biofilms that develop. And as I said, the beneficial thing here is that the biofilm develops on the surface of the fibers. The gases are depleted while they diffuse through the biofilm. And this is especially valuable if we talk about hydrogen as one of the substrates, where the solubility of hydrogen is 1.6 milligrams per liter, which is way lower compared, for instance, to carbon dioxide. You can actually buy these membrane bundles, and they are rather robust, as you will see. So this is, if it plays. So now it should work. So you see that they are rather stable. This is a movie from the company, and you can basically do what you want. They will remain stable over time. You can also autoclave these membranes so you can sterilize them. And in this video, they show even more variations of testing the stability of these fibers. So in Carmen, my PhD student was able to bring these fibers into these microfluidic chips, and she did OCT imaging, and you see how the biofilm grows on the surface of these fibers, and it gets thicker and thicker and thicker. At a certain time point, mass diffusion limitation will actually limit the thickness of the biofilm. And in BioOctane, our project, we will use this biofilm in order to feed an organism with hydrogen and CO2 as substrate, and the organism will produce 2,3-butane-diol as valuable platform chemical from hydrogen and CO2, and we will also use other waste streams in order to feed the organism for the production of these compounds. The general benefit is that we can supply hydrogen and CO2 via the membrane, and this gives a selective pressure for us as operators of these reactors so that the organism sticks to the surface because it needs the hydrogen and CO2. And we have oxygen coming from the bulk phase. So this means that we can grow an organism which usually has to grow with knallgas, so a mixture of hydrogen, CO2, and oxygen, but we do not use knallgas, because there's a spatial separation of hydrogen and CO2 from the oxygen. And this means that we can have a safe operation of these organisms that grow on knallgas, even also on larger scale. And in fact, another thing that we learned with these reactor systems is that Cupriavidus, the organism that we apply here, grows in these biofilms to cell densities that are tenfold higher compared to what this organism can achieve in planktonic systems. This is a very nice example why we should use biofilm systems, because it's the natural life form of the microorganisms. And because it's a natural life form of the organism, they grow to way higher cell densities compared to planktonic systems. And if you have more cells in the reactor system, you can get more product over time. Last but not least, last example are so-called bioelectrosynthesis systems. So systems in which the organisms sit on the cathode of a bioelectrochemical system, and we supply the organisms directly with electrons from the cathode, and the organisms use it as energy and electron source. And some of these organisms are autotrophic, so they use CO2 as their carbon source and produce, for instance, biomass or other compounds. And we isolated a very promising organism, which is called Coppelia spaumannii, that grows well on these cathode surfaces. What you see here is the relative volume, the biofilm volume over time growing on the surface of these electrodes. And we supplied either a potential of minus 375 millivolts versus standard hydrogen electrode or minus 500. And you see the biofilm grows equally well. If the potential is a bit too high, if we go to 0 millivolts, then the energy that we supply to the organism is not sufficient. So the biofilm does not grow, but as soon as we switch back to minus 500, it will start to grow. Why should you do something like this? We have a process in which a biofilm grows with an electric current. We are able to sustainably produce electricity using wind, using water, and so on and so forth. And if we have an organism like this that grows with aid of electrical energy and CO2, we can actually build biomass, and this biomass can be a protein source for the food and feed sector. And in order to show that this can be a viable process, we teamed up with Marvin Schatzinger and Martin Kalschmidt and tried to calculate the energy efficiency of these processes. And we compared the growth of our organism Coppelia spaumannii on the surface of the electrode with, for instance, photosynthesis by C3 plants, photosynthesis by C4, or microalgae reactors. And what you see is, if you compare this to photosynthesis, we are in the same area of solar energy demand for the process, but we can operate the process 24/7. We are not dependent on light. We are only dependent on electrical energy. Moreover, if we look at the efficiency, so the solar energy demand over the days of the experiment, so this is the average. But if we go through the process over the days, you will see that there is a time window within the process where we can even operate better compared to microalgae or photosynthesis in C4 plants. And this is when the cathode area is fully covered, but the biofilm is not already too thick. So our next aim will be to bring this, to scale up this process, and at the same time try to limit biofilm growth to this area where we can actually have the lowest solar energy demand. Here, the organisms grow with oxygen as electron acceptor. In other systems, we can grow, for instance, methanogens on cathodes. And these methanogens will use the CO2, which comes, for instance, from the off gas of a biogas reactor. Then you will use the electrons from the cathode and then convert also this CO2 into methane. So in the end, we will upgrade simple biogas, which always is a mixture of methane and CO2, to a biogas which consists only of methane to 100%. And we have examples for this that show that if you operate these reactors with organisms that can grow as biofilms, you will have a very efficient process. And these are electron microscopic images of 4 different methanogens that grow on the surface of the same type of electrode. And what you see by eye is that organism C and organism D is way more efficient in building a biofilm on the electrode structure, while the other 2 organisms rather grow only as thin biofilms, but also as planktonic cells. And when we compare the coulombic efficiency, so the amount of electrons that go into the end product methane, we see that these 2 cultures that have the highest coulombic efficiency are also the organisms that grow in the form of biofilms. All right, last but not least, a future look outwards into applied biofilm research and growth of microorganisms on electrodes. Together with my colleague Markus Ruttner from TU Hamburg, our aim is to grow biofilms on steel surfaces and to use these steel surfaces as cathodes And these biofilms will then build a surface coverage, a functionalization of the steel. And we have the working hypothesis that this will prevent corrosion of these steel systems. And these biofilms that grow on these, for instance, steel surfaces can be very hard. They can reach Young's moduli up to concrete. So our aim is to build three-dimensional biofilms on steel surfaces and to make them so hard that we have a very sustainable anti-corrosion agent in place. And with this, I would only like to remind you on the benefits of operating with biofilms. But I will also say that we are in the beginning of bioengineering in this regard. We need new reactor systems. We have to understand how we can bring into a reactor as much surface area as possible in order to grow the organisms. And we have to understand how long-term stable these production organisms are in the future. And with this, I would like to thank you for your attention. And I would also like to thank a number of people. Most of the work that I showed you is from Carmen, Leonie, René, Edina, Laura, and Miri. And I'm also happy that we get funding from the German Research Foundation, the German Ministry of Science and Education, and of course, by the European Union. Thank you very much.
Thank you very much, Professor Gescher. That was, for me, absolutely not being familiar with biotechnology in general, a very interesting deep dive into what seemed to me a completely new world. So thank you very much for that. Just a little remark, if you have any questions, you can write them either in the chat or via use the Q&A function. So we'll be happy to discuss them in the remaining 15 to maybe 20 minutes. And while you're like structuring your mind and arranging the first questions, I would have one which is more a general one, since you mentioned that more or less one of the next steps could be to scale up or to grow the size of the bioreactors. I remember you showed the one from the project, I suppose it was in Hanover, which had 100-liter size. So what do you see as current challenges towards scaling up bioreactors?
So I guess the biggest challenge is that we are not really familiar with these systems. So we operate in biotechnology, we operate with stirred tank reactors, and we do this for almost 100 years. And these biofilm reactors are something like a novelty, and we really have to understand how the organisms function in these systems. And as I said, there are a number of challenges. So challenges like how to get the system sterile and also long-term sterile, how to bring in as much surface area as possible with a stirable fluidic regime running over these systems, how to engineer the organisms so that they do what they are supposed to do over weeks or months of operation. And these are research questions that we would like to at least in part answer in the upcoming years.
Oh, yeah, very interesting. Thank you. What seemed to me as an important point is the thickness of this biofilm. You mentioned that like when it's too thin, it is not as productive as if it has a more or less ideal thickness, and when it gets too thick, it's also again disadvantages. So are there any ways to control the thickness of that biofilm?
Yeah. Yeah, very good observation. Thickness is a very critical point. If the biofilm gets too thick, we have mass diffusion limitations. So the substrate from the bulk phase cannot penetrate all the way into the biofilm. And also, if we operate with active substrata, it's counter-diffusionally the same thing. There are ways. We are also developing ways. My PhD student, Leonie, came up with a very nice idea. If we grow biofilms on cathodes, what she does is for a couple of seconds, she applies a very negative potential. And what will happen is that she intentionally forms hydrogen bubbles on the surface of the electrodes. And these hydrogen bubbles will actually lead to a detachment of these biofilm flocks, and then we can have a regrowth on the surface. My student Janek developed a trimmer that we can operate, that we can use in these rotating disc reactors, which does something similar. It tries to trim the biofilm to a certain thickness.
Oh, okay, interesting. So you either use like a sawtooth function and from time to time to reduce the thickness drastically, or have a trimmer even inside those small reactors. Yeah. Related to that, there's a question from the audience asking if the evolution in the biofilms is a good thing or rather a problem. So the assumption is that biofilms will adapt to consume and probably consume the substrate faster, and then they produce less than expected.
Yeah, this is a good question. So there are processes which are deemed to be used with biofilms, in which the organisms have a benefit from exactly doing what we want them to do. An example is methanogenesis. The organisms convert CO2 and hydrogen to methane, and they have a benefit from doing this because this is their supply of energy. And if we have these organisms operating in a biofilm system over weeks and months, these biofilm systems will get better and better and better because the organisms have a benefit from what they are doing. In other biofilm systems, when we basically force the organisms to do something which is not beneficial with regards to energy production for the organisms themselves, we actually have to invest in measures in genetic engineering of the organisms so that they cannot accumulate mutations that will lead to a failure in production.
Mm-hmm. All right, thank you. Since you mentioned genetic modification, that was another question that came to my mind. Where do you get the organisms from? So are they available somewhere? Do you need to modify them? If so, at which efforts do you need?
Yeah, we do both. So there are organisms where we use mixtures of organisms that occur in the environment. And there is this central dogma in microbiology which says everything is everywhere. It just depends on the process conditions what you will see. So, for instance, if we operate a bioelectrochemical system for a long time, we will see that organisms that have a benefit from growing on anodes will actually also be present mostly on the anodes. So we can operate these systems with naturally occurring strains, but at the same time, whenever we would like to produce something, especially besides, for instance, methane, we take model organisms where there is a genetic system, and we can actually change the genetics of the organisms so that they produce what we want them to produce.
Okay, interesting. You mentioned that the microorganisms can produce what you want them to produce, and there was one further question about selectivity. So as far as I understood, you have very clear inputs, for example, hydrogen or CO2. But what about the products of that conversion? So how is the selectivity? Is it always just one molecule, for example, methane, or is it more a range?
Yeah, that also depends on the organisms. Methane is a very good example because we usually then you only get methane as gaseous end product. With regards to, for instance, the organisms that we use in the BIOCTANE project, in which we teach the organism using genetic tools to produce butanediol, the organism is not only producing butanediol, but in this case also another intermediate. And we are trying to find ways in order to teach the organism only to produce the desired end products, which we can also do using genetic tools.
Okay, interesting. So a lot of range for adjustments as well here. Yeah. Another person from the audience asked about the memory effect that you'd shown from the lab experiments where you had the data from. So could you elaborate a bit on this memory that the biofilms showed there?
Yeah, we call it memory effect. This is a term that we invented. So what we see is if we grow, so I guess we have to see it from a certain size. Imagine a tree on the mountain. You will see on the shape of the tree where the direction of the wind, right? It will bend into the opposite direction from which the wind comes. And this is a memory effect because at a certain point, the tree will have this shape irrespective whether there is wind or not. So, and we think the same thing can also happen with biofilms. If we apply a certain shear force to the biofilm over a longer time period, the organisms will adapt to this shear force. They will form a less porous, denser structure. And if we then change the process condition, they will remain in this shape. And it will take a couple of days until the organisms learn this. But when they have learned it and when they have built this house will be stable over a long time period.
Okay, interesting. Yeah. More broadly speaking, since you mentioned that evolutionary process, can you give any indications on the timescales we're talking about?
So, for instance, with this organism Coppelia spormanii, which I was showing that grows and builds biomass on the surface of a cathode, we did an experiment. So we isolated these organisms from a volcanic source on the Azores, and the organism never grew before in its life on a cathode surface. So, and then we took it and put it on a cathode, and we inoculated this strain. So we incubated the strain for 4 months on this electrode. And then we sequenced what we had in the beginning and what we had in the end. And there were more than, if I remember correctly, more than 400 mutations that we saw overall in the community. And there were 10 mutations that we saw in all organisms growing on the cathode. And these mutations actually showed us what is beneficial for the organisms to grow under these process conditions.
Okay, interesting. So at least to me, being absolutely no expert, it seems like a lot of variation happening comparatively fast.
Yeah, that is true, because we operate with 10 to the 15, 16, 20 cells at the same time.
Yeah.
So this is the major difference to us. The number of cells, the frequency with which a mutation occurs is the same, but as we have way more organisms, we see way more mutations in a certain volume.
There's another question. I hope I get it properly. So have you thought about GeoBacter to make wires to facilitate that electron transmission through the cathode or anode within the biofilm?
We have been the current data that I showed on the anode surfaces, that was a co-culture also containing GeoBacter. GeoBacter species are known in the literature for being the most efficient with regards to anodic electron transfer. It would actually be a cool idea to use their conductive filaments and also use them in cathodic systems, which is something we could do in the future. Yeah, good idea.
Okay. All right, thank you. Related to that is one last question I received earlier on about the thickness of the biofilm again. So if I understood you correctly, you could also expect losses, for example, in terms of efficiency for a very thick biofilm, because the electron transfer, like depending on what organisms you use, etc., there's some sort of resistance throughout that film.
Yeah, that is true. Yeah.
Okay. As far as I see it, there are no further.
I see one raised hand on the attendees. Have you seen this?
Yes, I noted it. Let me see. Okay, we can try.
Oh yeah.
Miguel Petera, do you have a question? Okay, apparently it was just an old hen. Maybe he also asked the question in the chat then. If not, you can still type the question in. As far as I see it, there are no further questions though, and we have almost reached the end of this webinar. Okay. Thank you for that clarification. So I would like to thank you first of you all, you Professor Gescha, for this very insightful talk, and for me it was the journey into the unknown. Thank you very much. I think I learned a lot, and I'm pretty sure our audience also learned very much. And I would like to thank you, all of you as well, for being here for this BIOCTANE webinar. I hope you enjoyed this hour with the BIOCTANE project. And if this is the case, you can see us the next time on the 25th of April at 4 p.m. again, and we will also be sharing an invitation and the registration form, etc., on our social media channels. Or if you haven't already done so, you can also subscribe to our newsletter, which will also share the webinar invitations with you in due time. Having said that, thank you very much. I wish you a nice and pleasant afternoon, and see you soon.
Thank you.